All right, hello, 11s. How are you doing out there in internet land? We're almost done. Our unit on cell biology of cells. We're cellular. What cells do? So last time we talked about was harnessing the energy of nutrients. So the big important thing is gathering up nutrients, and then we learned that prokaryotic cells, they turn glucose into 2 ATP by a process called glycolysis. Eukaryotic cells, which have those membrane-bound organelles, then are able to further that and pump out 38 ATP in total, do it using cellular respiration in the mitochondria, using aerobic respiration in the mitochondria. This process, however, requires oxygen to be there. But one glucose molecule in eukaryotic cells produces 38 ATP. In prokaryotic cells, only 2 ATP. But those eukaryotic cells need oxygen because they're obligate aerobes, whereas prokaryotic cells that don't need oxygen are obligate anaerobes. And there's also some eukaryotic cells that are facultative anaerobes, which means that if there is no oxygen, they can start still producing ATP. Not as much ATP, but this is called fermentation. So yeast can make alcohols for beer, fruit can make wine and other liqueurs, and our muscles make lactic acid, which we then aerobically respire later when we've got oxygen again for ATP. Okay, so today we're going to talk about other cell specializations. What are some other things that cells do? So the main thing is they want to get nutrients, but they get those nutrients so they can do other stuff. So what are the other things that these cells do? So one of the things is protein synthesis. And that means making proteins. And remember, proteins are really important. Cells are made out of proteins. Proteins do all kinds of stuff in cells. So all cells need to synthesize proteins. So proteins are used as enzymes, and enzymes put things together or cut things up. So actually this reaction could go the other way and this way. So enzymes, I don't know why this one looks like a bat, but anyways, it doesn't matter. So enzymes take something, they cut it into 2 smaller pieces. So for example, this could be a piece of table sugar, and it cuts it into glucose and fructose, and then the glucose can go and it can be used for energy. So that's what enzymes do. They cut things, they put things together, they build up parts of the cell, they do all kinds of stuff. You find proteins also in the membrane. So there's all these things that proteins can do. They can let things in. They can cut things up at the membrane. They can do all sorts of stuff. Cell surface receptor. So they identify certain cells. You don't have to know the details of all these things. Just know that they're in the membrane. They make up parts of the membrane. They help move things in and out of the cell, act as antigens on the cell surface. So they're like chemical markers that identify cells. You can also have proteins that are hormones. Hormones, very important. Hormones travel around the body and they activate parts of the DNA in their target cells. So all of a sudden they activate your muscle cells to be more faster. So that could be adrenaline. What else? What other example of hormones? There's hormones that activate, for men, hormones, testosterone activates. Here, so you get an Adam's apple as you grow, as you go through puberty, or hair in your armpits. Hormones do all kinds of stuff. And there are proteins that make up structure. So the cell's cytoskeleton, remember we talked about that cytoskeleton, which holds the cell together, is sort of like the scaffolding inside the cell. Well, that's made out of proteins. Other things that, other structures that they make, well, like fingernails are made out of protein, a kind of protein. The connective tissue in between your muscles is made out of protein. So They're also used for structure. Now, what are proteins made out of? So we're getting to protein synthesis. We're getting to the details of that. So proteins are chains of amino acids. So all these amino acids come together to make a protein. Do I have a picture of a chain of amino acids? I don't, but that's okay, as long as you know that they're chains of amino acids. There are 20 amino acids, which means that there are countless combinations of these chains that you can make. And these chains can be 2 amino acids to hundreds of amino acids. The proteins can be tiny, tiny peptide chains, or huge, complicated, complex, they make complex shapes. Guess what determines the sequence of amino acids? What determines everything as we've been going through? What determines the proteins in the membrane, the proteins and enzymes inside the cytoplasm? What is it? This would be the part where I ask you, and you would all sit here quietly behind your masks looking at the desk. It's DNA. So DNA determines the sequence of amino acids. So it determines the structure and the function of the protein being made. And so the DNA sequence is kind of transcribed by RNA, and then the RNA goes out of the nucleus, and in the cytoplasm, the sequence of amino acids is read, and proteins are assembled at ribosomes. So remember, all cells need ribosomes because for protein synthesis, you need ribosomes to make proteins. Now. So that's protein synthesis. That's one of the things. That's a big thing that cells need to do. They need energy for that. That's why they need to eat things, gather nutrients. There we go. So now what happens once they've made the protein? They have to release the protein. So this is called the Secretory Pathway. And so proteins are synthesized in the rough endoplasmic reticulum. That's kind of this thing. It's this double membraned kind of thing that's right outside of the nucleus. And it's got ribosomes in it. Those little yellow dots are ribosomes. So proteins are made in the rough endoplasmic reticulum. There's also the smooth endoplasmic reticulum, which makes steroids. And steroids are like fats. And the smooth endoplasmic reticulum looks like this, except it doesn't have the ribosomes, because ribosomes aren't necessary to make fats. But that's good. We're starting with this. So either you get steroids, which are hormones which go out of the cell and they activate other parts of the cell and do all kinds of amazing stuff, adrenaline and norepinephrine, make you happy, make you sad, all these kinds of things. So the steroids will be made in the smooth ER. This one is showing the rough ER, so this would be proteins being made. And they could be enzymes that need to go out of the cell and do something. Maybe there's some danger outside of the cell, so your body needs to quickly make a protein that goes and kills the danger that's outside, breaks it up, the danger. So it makes an enzyme. So it will make this, the DNA sequence will... The DNA sequence will be transcribed in the nucleus somewhere over here behind this writing, and then the RNA comes out of the nucleus, and it's translated into a peptide chain. So this isn't the fully assembled protein yet. And then that peptide chain they fill up inside the rough ER, and the rough ER blebs. It's called blebbing. It blebs into what's called a transition vesicle. And so this vesicle, it's just basically a membrane bubble full of that peptide chain, that protein, and it just travels to this thing called the Golgi apparatus. And in the Golgi, what happens is those peptide chains will get changed, will get ready to go out of the cell. So maybe they have to join another peptide chain to become a fully operational protein. Maybe some part of it has to be cut off to make it active, and the cell doesn't want it active too early, because if it's a thing that eats stuff, the cell doesn't want that thing near the nucleus eating things just by accident. So it kind of makes the protein not work. And then in the Golgi, the protein gets modified to get ready to work. Same with steroids. The steroids also will make it to the Golgi, and they might get modified, or they might not. They just go to the Golgi and kind of make their way through the Golgi. But the Golgi modifies and get things ready for excretion. So then it travels to the Golgi, the changes are made, and then it gets packaged, a piece of the Golgi blebs off, and then you get a secretory vesicle. And that vesicle then travels to the outside, the plasma membrane, the membrane of the cell, and it fuses. It fuses itself with the membrane, and if you imagine, and then it just dumps the contents out of the cell. So that enzyme then can go and kill whatever danger that is outside of the cell. Those vesicles and those Golgi bodies, remember, these are membrane-bound organelles. So this will be in eukaryotic cells that this is happening. It's not in prokaryotic cells, the secretory pathway. And they're made out of membrane. So when those vesicles fuse, they're just fusing with more of the vesicles are made out of membrane as well. And then they contain this stuff in there. So that's an important thing to note. And they're not made out of something special. They're just made out of membrane. And the membrane is those phospholipids, that double layer of phospholipids. So that's a thing. That's another thing that cells do. That's another cell specialization, and it's a big one because we have lots of cells. There's that's all the cells in your cheek. There's cells in your cheese. There's salivary glands. There's cells in your cheek, and they do this constantly. When they detect that you've got food inside your mouth, they package an enzyme called amylase. So amylase starts getting made, and it gets packaged just like this. It goes through this whole thing in the Golgi, and it gets dumped out. And amylase dissolves starch and sugars. It starts to break them down. So already in your mouth, it starts to break that food down. So when it makes it down your body, it's already, a lot of it is broken, and you can get that sugar, those yummies right away into your blood. Okay, so that's the secretory pathway. And another thing is intracellular digestion using lysosomes. So this is, important thing, this fusing and dumping is called exocytosis. Exocytosis. It's a type of phagocytosis where the membranes fuse, And exocytosis means things go out of the cell. Here, intracellular digestion, what happens is you have, again, phagocytosis, but it's called endocytosis, means things are going in. And this is a cell eating. So this could be a single-celled organism, or it could be just an animal cell. And so what happens is the membrane, the whatever it is, the membrane kind of envelops the food, and then it turns it into a vesicle, okay, or a food vacuole. And the vacuole will be full of the extracellular fluid and the food. And then what happens is something called lysosomes, which are just kind of specialized vesicles. Again, all these are just bubbles of membrane, and they are full of protein. They've gone through this whole process, except they haven't merged with the plasma membrane. These lysosomes are just this, but they're floating around in the cytoplasm. And what they do, instead of merging with the plasma membrane, they merge with these lysosomes, with these food vacuoles. The lysosomes merge. They do the same thing. They join up and open up with these food vacuoles, and then those proteins, or those enzymes and proteins that are in there, they digest the food, so they break it up into little bits, and those little bits get transported out now into the cell. So it would be sugars and stuff that the cell, or whatever it is that the cell needs, amino acids, nucleic acids, fats, or lipids, whatever it is, it will get transported out, and then the cell can use it, and the garbage will then, the food vacuole will merge with the plasma membrane and do exocytosis, another form. So it'll get dumped out. Basically, that's the cell pooping. So that is intracellular digestion. And this is called phagocytosis, is when cells eat. Phagocytosis, when they bring in something big. Pinocytosis is when they bring in something really small, something tiny gets brought in, usually liquid. So phagocytosis is called eating. Pinocytosis is called cell drinking. Okay, so that's another cell specialization that cells can do. They can eat and devour. That's how they eat. Now you know how they eat. Isn't that amazing? Okay, next. This is a big topic, so this video might be a little bit longer, but we have to finish it. So cell reproduction. So cells wear out. They get older. They do all that metabolism. They're doing stuff, and just like people, you know, Mr. K is getting older. He's a lot tireder. If I was making this video 20 years ago, it would be a lot funnier and smarter and probably quicker and take less time because all the things in my brain and stuff worked better. Now I'm just a little bit slow and a little bit tired and behind the ball. My knee hurts and things. All these things are happening. And it's the same thing for cells. As they get older, all the things wear out. So what they do is they make themselves new again. They reproduce. And there's two main types of reproduction. There's asexual reproduction and sexual reproduction. Asexual reproduction, so single-celled organisms can carry out asexual reproduction. And so prokaryotes like bacteria, that's what they do. They eat, they get bigger, they get worn out, and then it is time for them to reproduce. And what they use is this process called mitosis. And what happens in mitosis is all of their DNA gets doubled up. DNA replication. So it gets replicated, it gets copied. So then you have two copies. These are two chromosomes there. And they line up the chromosomes. This bacteria or this cell has two chromosomes. And so each V, if you look at the sideways V's, is a copy. Each V is a copy. So this cell started out with two chromosomes, and then it copied its chromosomes, and they're joined in the center. And then what happens is, so they line up, and the cytoskeleton has a lot to do with this lining up and this splitting, and then the cell splits, and one copy of each chromosome stays behind, and then you have two daughter cells with identical, so that they have identical copies of the two chromosomes. Now, one thing we say they're identical copies, but actually we know that they're not identical, because every time you copy DNA, you make little mistakes. But we'll talk about that in the evolution section. But for now, those are identical copies, barring any major mutations. That's what those are called, is mistakes in the DNA. And so, very simple. And this method, bacteria use this. They could use this when times are good. There's lots of sugar. There's lots of stuff for them to eat. Like say you've got that awful flesh-eating bacteria, and so it's living in the tissue of your flesh, and it's eating your skin, your flesh, those tissues. It is so happy. It is doing mitosis. It's dividing. It's consuming your flesh. You find you've got this sore that won't heal, and this growth is exponential. So one cell makes two make four make eight make 16, 32, 64, 128, 256, 512, 10,000, or 1,028, 2,056, and on and on and on. You see it goes up and up and up and up. It's exponential growth. So now the sore is going to get bigger and bigger and bigger. And then that's the advantage of this asexual reproduction, that it's very fast. You can make a lot. So prokaryotes really like to reproduce this way. Can produce a lot quickly. And then sometimes there's nothing you can do. They have to cut off your leg or whatever part of you it is that has these bacteria growing on it. So don't get flesh-eating disease. That's it's a bad one. Now, the other way of sexual reproduction, you see it's more complicated, is sexual reproduction. And this is how we reproduce. We use sexual reproduction. Now, bacteria, prokaryotes, also can do sexual reproduction. I'll talk about it in a second. And so how this process works, the process called meiosis. And in meiosis, you basically get mitosis. You kind of copy first two cells, and then those cells, those two daughter cells, they split again, And so what ends up happening is then there are 4 sort of daughter cells, and those 4 daughter cells now only have half the chromosomes because you've only duplicated the chromosomes once. So you duplicate all the chromosomes. They kind of arrange themselves just as they did in that bit, and then the cell splits, so there's 2 copies of the chromosomes. But then it splits again, so only half the chromosomes make it. And the chromosomes are independently assorted. So what does that mean? Well, all of us have, we have the genes from our mom and from our dad. So each chromosome has 2 copies. And 1 copy is from your mom, 1 copy is from your dad. And so, for example, I'll use my kids as an example. I have brown eyes, my wife has blue eyes. So one of my daughters has blue eyes. What that means is that my wife's egg, which only carried half her chromosomes, had the gene on one of the chromosomes that carries blue eyes. I carry chromosomes from my mom and my dad, and it just happens that all my genes, you carry 2 copies of each of your genes, 1 from your dad, 1 from your mom. One of my genes is a blue-eyed one, and one of them is a brown-eyed one. And I have brown eyes because if you have blue and brown genes, if you have the 2 genes, the complementary genes, you will express brown because it's the dominant gene. So when my sperm was made, that resulted in my first daughter, when I underwent meiosis and only half my DNA made it, only that blue-eye gene made it into that sperm. And then my wife has blue eyes, so she only has the gene for blue eyes. So when it met with the egg, it was blue, and we have a daughter with blue eyes. Another sperm that made my other daughter, though, got the brown gene, and then the brown gene met my wife's egg, which had the blue gene, and my other daughter has brown eyes because brown is dominant. So you could see how this method is more complicated. Even that explanation was terrible. I don't think it made any sense. It didn't make sense to me, so I'm sorry about that. But basically, the thing that you get from this is it's more complicated, and what happens is there's more shuffling. So in mitosis, you have identical daughters, right? But in sexual reproduction, all the genes, the chromosomes get shuffled around. There's chromosomes from the mom, there's chromosomes from the dad, and random ones get selected to make the offspring. So the offspring are a random combination of the parents, not an identical copy. They're a random combination, so they're going to be different. In mitosis, there's no other organism, there's no other parent that is making in asexual reproduction, there's no other organism that's adding genes. In meiosis, it takes 2 members of the species to make offspring. So these daughter cells, these are called gametes, then will meet gametes from another organism. They will mix, so they will have a full complement of chromosomes. And because these only have the gametes, so this will be sperm or egg, only have half. Now, I said, so prokaryotes, what they do, bacteria, they do sexual reproduction as well. They will copy their genes and then send some of them to another bacteria. It's called conjugation. They do this when times are tough. So if you've got that flesh-eating disease and you start treating it with antibiotics, and it starts killing the bacteria. If that antibiotic is really good, it targets some specific protein or some specific thing, some specific cell function that those bacteria do. If all the daughter cells are the same, they will all get killed because they're all the same. But if they can rearrange their genes, if they can mutate fast enough, they might be able to beat the antibiotic. So when times are tough, when bacteria are starting to die, they switch to conjugation so that they can rearrange their genes, they can resort their genes. That's the advantage of sexual reproduction, is you combine your resources, you combine your adaptations. That feels good. It's going to come up in evolution as well. Okay, the last thing, it's a tiny thing, is unicellular versus multicellular organism. The big advantage to being multicellular is, well, you have places, you have specializations, you have tissues and things, and the cells can differentiate themselves. You got a lot more complexity, so you can store food. You can go without food for a long time because you can store food on your body. You can make cells that purely store food. A tiny unicellular organism needs to get food all the time to feed its metabolism because it's got nowhere to store that food. The other thing is tiny little organisms are really susceptible to temperature. Water, because of the properties of water, it absorbs temperature. It either keeps its heat or you can put heat into it and it won't heat up too much. And our enzymes and proteins only work in a small temperature range, about 36.5 degrees centigrade. So if it gets too cold, there's not enough energy to do metabolism. If things get too hot, our enzymes actually and proteins start to fall apart. So a large organism is actually a large body of water. There's a lot of water in our bodies, so it maintains temperature very well. We can also have specializations that keep temperature steady. So we sweat and we sweat to cool down. We can shiver to heat up, all these things. Whereas unicellular organisms, they can't. So they're very susceptible to changes in temperature. That's a big one. The disadvantage, though, that we have is we need a lot more food when we do get food because we're much larger, so we have greater energy requirement, and we've got a lot more complexity. I don't know how that's a disadvantage, but I guess, if you had to fix, it's a lot, I guess, like if you look at what's going extinct on Earth, it's these large mammals, right? So like elephants and hippopotamuses and rhinoceros and large sort of complex animals are not as resilient. So when you have lots and lots of tiny unicellular organisms, yes, you can murder them in huge droves, but there's so many of them that it's hard to kill them because they're less complex, so they're easier to make. You can make so many of them because they're not as complex. So there's the disadvantage to being a multicellular organism. Okay, there you go. That's it. We're done. Unit B.